Okay, good morning. My name is Tirtha Sivanandam, and my mentor is Dr. Natalie DeForest. Let me begin with the paradox. About 3 out of 4 patients who experience a myocardial infarction or heart attack have cholesterol levels that fall within the normal range. If cholesterol screening is meant to identify cardiovascular risk, why do the majority of heart attacks occur in individuals labeled low risk? This paradox motivated my research. Okay, there. To address this, we need to distinguish between different types of cholesterol. The first one is LDL cholesterol, or low density lipoprotein. It's often called the bad cholesterol, and HDL cholesterol, or high density lipoprotein. This is considered the good cholesterol. However, there is another cholesterol particle known as lipoprotein A, or LPA. Structurally, it resembles LDL, but has an additional protein attached called ApoA and ApoB100. This makes it particularly atherogenic and prothrombotic. Crucially, LPA levels are more than 90% genetically determined and are not significantly altered by diet, exercise, or statin therapy. Okay. Myocardial infarction remains the leading cause of death globally, with nearly 9 million deaths annually. Despite reliance on LDL, HDL, and triglycerides for risk assessment, these conventional markers fail to explain why the majority of infarctions occur in people with normal LDL levels. This diagnostic blind spot raises the possibility that LPA is the hidden driver of risk in these patients. My research question for today is, does genetically elevated LPA mediated by the LPA gene variants casually and independently increase the risk of myocardial infarction in individuals with normal cholesterol? And my hypothesis is that LPA variants elevate LPA, which then increases infarction risk through both atherogenic and thrombotic mechanisms, independent of LDL cholesterol. This study had 3 objectives. The first one was to elevate the association between LPA and myocardial infarction in individuals with LDL less than 130 mg by milligrams by deciliters. The second is to test whether LPA is causal using Mendelian randomization, and finally, to assess whether LPA provides additional predictive value beyond standard lipid measures. Okay, so the research design. We used 2 data sources from 2 publicly available sites. First was the GWAS catalog, and second was the NHANES catalog, which included studies from 2017 and 2018 and the 2021 to 2023 cycles. 2019 and 2020 was excluded due to COVID, and then there was a lot of data limitation which happened around that time period. I included adults aged from 30 to 75 years with LDL cholesterol below 130 milligrams per deciliter, which is considered low risk. Participants were required to have complete data for LPA, myocardial infarction status, and covariates. The rationale for setting the LDL cutoff at less than 130 milligrams per deciliter was to isolate a normal cholesterolemic population. This reduces confounding from overt hyperlipidemia, meaning cases where extremely high LDL would be the obvious case of the infarction, and allows the analysis to focus on hidden risk driven specifically by LPA. Okay, the observational analysis included a multivariable logistic regression. Logistic regression is used when the outcome is binary. Here, the outcome was history of myocardial infarction, yes or no. The model estimates the log odds of the outcomes as a function of predictors. When exponentiated, these coefficients yield odd ratios, which quantify how much the odds of the infarction change with each unit increase in a predictor. The exposure was plasma LPA, modeled as a continuous variable and log transform for normality. The outcome was self-reported history of myocardial infarction, and the covariates included age, gender, LDL, HDL, cholesterol, hypertension, and smoking status. I also calculated variance inflation factors, or VIFs, to assess collinearity among lipid variables. VIFs were low, indicating no multicollinearity and confirming model stability. The second method was Mendelian randomization, or MR. Mendelian randomization uses genetic variance as Mendelian randomization uses genetic variants as instrumental variables to infer causality. Because alleles are randomly assigned at conception, genetic variants are not confounded by lifestyle or environment as they precede disease onset. Thus, MR approximates the logic of randomized controlled trial. For this study, I used two well-characterized LPA SNPs, RS3798220 and RS104555872. SNPs, or single nucleotide polymorphisms, are the most common type of genetic variation in the human genome. A SNP represents a single base change at the specific position in DNA. These small differences can affect gene function. In the case of the LPA gene, certain SNPs, such as RS3798220 and RS104555872, increase lipoprotein levels in the blood. Because SNPs are inherited randomly at conception and remain fixed for life, they can be used as instrumental variables in Mendelian randomization to test causal relationships between biomarkers and disease. The exposure effects variant to LPA were derived from genome-wide association studies. The outcome effects variant to myocardial infarction were obtained from large-scale GWAS meta-analysis of coronary artery disease and myocardial infarction. The Wald ratio was applied per SNP. Robustness was established by the concordance of results across both instruments. The final sample of sorry, I must have gone to the slide before this, but the observational analysis and the final sample consisted of 1,562 adults with LDL less than 130 milligrams per deciliter. Results showed that each 1 unit increase in log transform LPA was associated with a 2.7 increase in the odds of myocardial infarction. LDL and total cholesterol were not significant predictors once LPA was included. Thus, LPA independently predicted myocardial infarction risk, while LDL did not capture risk in this group. Next, we have the Mendelian randomization. This table represents the Mendelian randomization results for both LPA variants. We first have the odds ratio. This is the causal effect estimate from the Wald ratio. For RS3798220, the odds ratio was 2.63, and for RS104555872, the odds ratio was 2.78. These indicate that carriers of either variant have more than a twofold increase in risk of myocardial infarction. We next have the 95% confidence interval. Both SNPs demonstrate extremely narrow confidence intervals, reflecting the high precision in the estimates. Our p-value, which for both are far below conventional significant thresholds, confirming that these results are not attributable to chance. The critical observation is the near identical odds ratio between both independent variants. This concordance provides robust evidence that the elevated LPA is a causal determinant of myocardial infarction, rather than just a correlated marker. These results converge on 3 conclusions. First, and then this is a little bit more on the Mendelian randomization. As you can see, we have the odds ratio as 2.63 and 2.78, which show that how much more of an increase someone with the SNP can have for myocardial infarction due to the LPA variant. Okay, and these results converge on 3 conclusions. First, LPA is an independent predictor of myocardial infarction. Second, genetically elevated LPA confers a more than twofold increased risk. And third, LDL does not explain infarction risk in normal cholesterolemic individuals. Together, these findings resolve the paradox of myocardial infarctions occurring in patients with normal cholesterol. Okay. Let me just go back for 1 second. Yeah. LPA represents a substantial hidden risk factor. Approximately 20 to 25% of individuals have elevated LPA, yet remain undetected under current LDL-focused screening. Furthermore, LPA levels vary by ancestry. The LPA gene contains a region called the KIV2 domain, or the Kringle IV type 2, which can be repeated a variable number of times. Individuals with fewer KIV2 repeats tend to have smaller ApoA isoforms and significantly higher LP levels, whereas individuals with more KIV2 repeats have larger isoforms and typically lower LPA levels. The distribution of these KIV2 repeats differ by ancestry. For example, African populations more commonly inherit the low repeat alleles, which results in higher median LPA concentrations compared to European or Asian populations. So, the difference is not due to environment or lifestyle. It reflects underlying genetic architecture in the LPA gene across populations. These findings support two major clinical implications. First, incorporating a one-time LPA measurement into cardiovascular risk assessment. This would identify high-risk individuals invisible to LDL-based testing. And second, leveraging emerging therapies, including antisense oligonucleotides and siRNA, that can lower LPA more than 80%. For the first time, we may be able to both detect and directly treat this casual risk factor. In conclusion, this study demonstrates that elevated LPA, driven by LPA variants, is a causal and independent driver of myocardial infarction in normal cholesterolemic individuals. Logistic regression established association, and Mendelian randomization confirmed causality. Both methods converge onto the same conclusion. LPA more than doubles risk, while LDL fails to capture it. By integrating LPA testing into routine practice and combining it with novel therapies, we have the potential to prevent millions of myocardial infarctions worldwide. This research serves as the first step towards closing that diagnostic gap. Thank you.
Thank you so much for sharing more about your research. And then again, as a reminder, if you have any questions, feel free to add those in the Q&A section of the chat. Again, that might be under the More tab in your Zoom. But to kind of get us started, I had some questions around LDL versus LPA. So it sounds like LDL is considered in the standard risk calculation whenever people are getting assessed, but not LPA. Do you know why that is?
So, LPA is determined by the LPA or lipoprotein A is really determined by the LPA gene, which makes it a little bit more harder to identify through the standard cholesterol testing. Right now, it's just mainly LDL and HDL because those are just determining the amount of cholesterol that enters to the arteries of the heart, whereas the lipoprotein A has to be determined by the LPA gene and the number of KIV2 repeats. So it tends to be a little bit more difficult to do, and that's why it hasn't been implemented as much as the other standardized ones, which is really important because there are about 20% of our entire population is at risk of having this LPA gene, and it goes undetected so often. That's why it's really important. And it's also that it's a little bit more expensive because obviously you have to go through the gene and then identify it. But it's it's really a debate whether it's like you're willing to spend that much money to save a life. So I think this research is really important in that way because it kind of opens a new door to identifying more ways to save people from these myocardial infarctions that happen without realizing that there is something, an underlying risk factor through our bodies that we might not realize it based on what we know from like the standardized testing.
Yeah, very interesting. And then I'm also curious, is the LPA gene involved in anything else besides cholesterol?
Not really. Also, mainly because since the LPA gene, it affects the lipoprotein A, and that generally has the same structure as the LDL. It just has the APO A and APO B 100. So as a result, this lipoprotein specifically only affects the heart, and it hasn't really been identified through anything else, especially because these SNPs from the casualty that I've read and just like tested out with the Mendelian randomization, it only directly affects the heart. But I'm sure that there are other genes that we haven't figured out yet that do affect in similar ways for other mechanisms of your body, like maybe the lungs or the stomach and things like that. So that would definitely be something that people should research into or something I'd be interested in. But I feel like this is something that is really important also because it's a little bit known and it still hasn't been really discovered into. So, yeah.
Great, perfect. Thank you so much.